experience. Why did this helpful? Yeah, I know that.
Yes, Dr. Tariq, please go ahead. You can see your slide. Tariq, please first let me share the screen and then you can on your mic.
Yeah, thank you.
Okay, we can hear you now. Please share
Hello
the screen.
everyone.
Yes, please go ahead.
Yeah, so I was talking about plant biotechnology as an important avenue for medicine during pandemics. I have a PhD in plant biotechnology, and me with my students and colleagues, we are working in plant biotechnology, developing medicine, different medicine through different plant biotechnology, plant in vitro cultures. So what we did was this collaborative effort on using plant biotechnology and bringing attention of the world towards plant biotechnology. That plant biotechnology could be very, very helpful in the case of different pandemics for future. As you know, COVID-19, which is caused by SARS coronavirus 2, of course, has alarmed the world of the diversity of threats surrounding us in the form of pathogens. Timely management of such pathogens and diseases they cause is extremely important. And again, there are vaccines available now, luckily, but vaccines are only preventive agents, and we need healing agents to treat such diseases and recover and save patients who are already suffering with diseases like coronavirus and any unfortunate future diseases. So the current treatment and prevention options explored include passive immunity, as Professor Shivani talked about, the repurposing of existing drugs like chloroquine, hydroxychloroquines, the different vaccine candidates that I'll be talking about in the next slide. Then the alleviating agents like dexamethasone, plant-based vaccines, those developed by Medicago, a Canadian biotechnology company, from Nicotiana vintamiana, and then plant-based medicine, such as those from the dried fruits of forestry practice. As you could see in this slide, there are currently 119 vaccine candidates, of which 33 are in stage 3 clinical trials, and so far 15 virus vaccines have been authorized in several countries by different authorities. The famous one, as you know, are those from Pfizer. These are the primary developers. You might not be able to clearly see this one because it's a small font, but the primary developers are Pfizer for PNT 162B2, and then Moderna vaccine, AstraZeneca, Sputnik, all the way to the CoronaVac developed by Sinovac, the BBIT CoronaVac by Sinopharm, different vaccines developed in Kazakhstan, Uzbekistan, Russia, Bharat Biotech, and all those vaccines developed in different organizations in different parts of the world. So 15 vaccines have been authorized for use. Now, as I already said, vaccines are preventive agents. What could plants do for such pandemics in the case of such diseases is to provide healing agents, and these plants provide these healing agents through secondary metabolites. As plants produce a diversity of organic compounds, the four major classes of these organic compounds, these secondary metabolites, what they call alkaloids, glycosides, phenolics, and terpenes. Several different plant-based compounds have been shown to be effective against previous types of the coronavirus, SARS coronavirus 2, which caused outbreaks in 2002, 2003. In case of that coronavirus, one of the studies published in Lancet, a compound called Glycyrrhizin was very effective against that SARS coronavirus, and we could just give or take a hint from that there is a diverse potential in secondary metabolites from plants. These compounds have been employed for their different mechanism of action, such as inhibition of replication, the stopping the entry of virus to the cell, and things like that. So why plant biotechnology? Plant biotechnology provides this really interesting area of in vitro cultures that provide an option for making the best use of plant machinery to provide medicinally important secondary metabolites. And these different cultures, as you could see in the figure, callus culture, adventitious roots culture, cell suspension culture, hairy roots culture, these are initiated from an explant that ultimately is derived from a plant grown in the lab, in vitro. And these different cultures are really the factories of medicine that could be used that could be upscaled in a form of bioreactors to produce really interesting different secondary metabolites that have the capability to heal people and to prevent mortality. One of the areas that plant biotechnology focuses on is not only to isolate medicinal compounds from these different cultures, but also to try to enhance or produce novel compounds. And that area, we call that area as elicitation of in vitro culture. So if you could see with this sort of complex looking figure that I constructed for one of our papers, is this that if from the in vitro seeds, we start to generate different plants in the laboratory in growth chambers, and we could use different modified light condition, continuous light condition, different chemicals. We could provide it with different chemicals, different nanoparticles or nanomaterials. We could manipulate them genetically. We could use enzymes as elicitors. All those different modifications sort of act as elicitors are stress-giving agents for these plants, and in the plants, in stress, these plants directly provoke their immune response or defense response to produce these compounds in bulk and higher quantities. We could then extract these compounds, identify them, characterize them, properly test them in different phases against different pathogenic agents such as the SARS coronavirus 2. And then another area that comes in line with plant biotechnology is is the metabolic engineering of plant, the production and enhancement of compounds like anti-coronavirus compound. For example, giving such sort of genetic transformation in certain genes, switching on or off different genes for activation of plant defense mechanism, or poor expression of certain enzymes or expression of new desired genes, so that we could produce novel compounds, or we could just enhance the production of existing compounds and then upscale those cultures, those plant parts or different roots or cells directly, or cell suspension of the plant to produce the product in bulk. Then another area, as I mentioned in the previous slide, is the transgenesis. Plants are genetically modified through different techniques as agrobacterium purification, particle bombardment, gene gun, electroporation, and different methods to modify these plants, produce transgenic seeds, and all the way producing different compounds to these plants, genetically modified plants. As you know, CRISPR-Cas is one of the technologies that is very, very famous for gene editing, and that could be one area to modify plants for the production of compounds of our interest. And lastly, one of the area we call as farming, producing vaccines in plants through different modified techniques. For example, in different plant parts like leaves or fruits of the plant, to try to develop vaccines, to try to develop antigens purified from plants that could be very much biocompatible, that could be very much safe to use as they are coming from plant origin and also have different ease of different regulatory approvals. That's all from my side. Thank you all for listening. I really appreciate the efforts of Pakistan Academy of Sciences and ASA for their efforts, and also I thank my students at the University of Malakand and my colleagues for their joint collaboration and efforts. We have already published some of the interesting papers in journals like Frontiers of Plant Sciences, and we are working closely, very keenly to develop certain compounds that could be helpful in this area. Thank you very much.